proteins are not little colored squares.
I'd like my slide number 29, please.
Good luck.
Good luck. Slide 29.
I want to recapitulate Jim's remark and then respond to it. He said, why are we having this debate at Biola in what he implied was a religious setting? Why isn't this debate going on in a scientific setting? And I'm waiting on my slide. While I'm waiting on it, I'll give you a story. In October last year, I was in Washington, D.C. To speak at the American Enterprise Institute, and I went to the American Association for the Advancement of Science building, which is the largest organization of scientists in the country, to hear a scientist from Caltech named Chris Adami. And the room was jammed, the auditorium was jammed, very much like this room is tonight. And Chris Adami was describing an experiment that he and Richard Lenski and some colleagues published in Nature, one of the very journals that Jim mentioned, next to Science, the most prestigious science journal in the world. This experiment, which I'm sorry, that's not slide 29. Could you give me slide 30, please? Yes, thank you. This is the experiment he was describing, published in Nature, a major paper. And that night, Chris said, I want to tell you why I began this research project. And he flashed up a slide of the cover of Darwin's Black Box. He said, Mike Behe has made an argument about the nature of living things, namely that they have these irreducibly complex properties. This is a fascinating argument, and I want to respond to it as a scientist. So he and Lenski and his colleagues wrote a computer program to try to evolve, via Darwinian mechanisms, apparently irreducibly complex instruction sets. Now, the AAAS videotaped that speech. It was clear that that night, Chris Adami's research was to respond to the scientific case that Mike Behe was making. In detail. The whole context of his research was to do that. And this paper, published in Nature, was to do that. Can you advance it one step? This is the opening sentence of the abstract. A long-standing challenge to evolutionary theory has been whether it can explain the origin of complex organismal features. Now notice the verb here. It's a passive. Who is raising that long-standing challenge? Well, it wasn't Chris Adami. It was Mike Behe. It was the people on this panel. Now, here's why this is weird. There's a funny kind of asymmetry with respect to intelligent design in the scientific literature. It's perfectly possible to critique intelligent design and to publish papers like this in Nature. But Mike Behe would have a very hard time getting one of his own papers in, making a case for irreducible complexity in the same journal. Now that, I think, is like playing a game of softball with two sets of rules, one for the home team, one for the visitors. The debate about intelligent design has been joined in the scientific literature. One of the papers that Bruce Weber put up from Science just a couple of weeks ago, Chris Adami wrote a commentary, a News and Views commentary about that paper. The title of that commentary was Reducible Complexity. And the whole argument of his commentary was, we now have evidence to answer Mike Behe's case about irreducible complexity. So this is the other most prestigious journal in the world, making a case against irreducible complexity. So you can see that a debate has been joined in the scientific literature about intelligent design. It's already going on, but there's this funny set of rules. One set of rules for the home team, one set of rules for the visitors. I look forward to the day when Mike can make his case in the same journals the way his critics can. Thank you.
I want to emphasize that it's real easy. There's kind of a rhetorical dissymmetry or asymmetry in this debate, in that Darwinists want you to think that making transitions is easy. So they'll kind of say, oh, well, that's no problem. Let's just start with a paperweight, and then we can go to a keychain. So it's not irreducible.
Michael?
But I would suggest, let me finish this sentence.
Just the only thing is that going from a paperweight to a keychain is not the same as going from a gene for one function to another, that maybe 5 mutations are sufficient.
Yeah, well, this wasn't my analogy. This was Ken Miller's analogy, and I just want to suggest that here's the problem. Not only is this, I would consider this an intellectually unserious response to the problem of irreducible complexity. Another problem is that if you ask yourself, if you start with some structure that's being selected to be a paperweight, it's not going to be prepared at all to be a keychain.
Okay.
A structure that's a keychain isn't going to be prepared at all for a clipboard. And if somebody says, I'm going to accept this and use it for a clipboard, well, you may as well say, I'm going to accept a paperweight. You haven't gone any of the way towards a clipboard.
But Michael, what about the question I asked?
Okay, I thought I was answering it. Now let me continue then. Could we go to the skip the next two slides to the third? This is kind of a slide that's similar to the one that Professor Weber showed. There we go. Thank you very much. And here's the idea. Suppose you have an irreducibly complex molecular machine made up of 6 different parts, labeled A to F. Suppose that they started out as individual precursors, each of which had their own function. Could you, next slide, please. Now suppose they got together somehow. Now you could have an irreducibly complex machine, the story goes, but each of the parts had their own function. And I submit that even if all of these things are doing the exact same thing they would do in the irreducibly complex machine, you still can't get from these parts to here, even if they're doing the same thing. And why is that? Well, here's one reason. Proteins are not little colored squares.
Exactly.
They are not little colored figures like Ken Miller showed on his slide. Why does the A go next to the B here, besides being in the alphabet there? Why doesn't it go down here? Why aren't these in a line instead of something like this? What makes a real protein stick to another real protein and another real protein stick to that? They stick to each other because they have very complex surfaces which are matched to each other. The chemical and physical properties have to be just so, like little magnets that are lined up in order to allow them to bind.
The challenge has not been met of a gradual account of the origin of those molecular machines. This hand-waving and referring to paperweights and stuff is all kind of cute, but it's a word game rather than an actual scientific explanation.
This is a cat on, well, acid. LSD.